I'm going to be talking about Nirenberg and Mattei's amino acid experiment, then later on I will talk about Nirenberg and Letter's experiment. For Nirenberg and Mattei's experiment, the question was asked, what amino acids are specified by codons composed of only one type of base? At this point, we are all familiar with what a codon is. It is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule. The letters that make up the codon determine the amino acid. Here's the coding dictionary that shows all of the possible codons and their corresponding amino acids. For the experiment, their mixtures included an artificial form of RNA consisting only of uracil called poly-U, a cell-free extract, and 20 amino acids. They used 20 test tubes, each for a different radioactive labeled amino acid. Upon experimenting with poly-U, the results showed newly synthesized proteins made up entirely of phenylalanine, equaling the codon UUU. This was evident because the newly formed protein was radioactive just like the one radioactive amino acid, indicating the protein was made up of that radioactive amino acid. This experiment was repeated with poly-A and poly-C, finding that lysine is AAA and proline is CCC. Poly-G did not work. So did Nirenberg and Mattei answer their question about which amino acids are coded with one type of base? They did. Now let's move on to Nirenberg and Letter's amino acid experiment. Here's the question they asked. With the use of tRNAs, what other matches between codon and amino acid could be determined? They used very short artificial RNA sequences called trinucleotides, ribosomes, 20 charged tRNAs with one amino acid being radioactive. In this process, the short RNA sequences allow the ribosome to bind with the complementary tRNA specific to that one trinucleotide or codon. They made one amino acid radioactive and ran the mixture through a filter. Ribosomes with bound codons stuck to the filter while the unbound ones filtered through. Those stuck to the filter paper made the paper radioactive like the amino acid, meaning the specific codon coded for the corresponding amino acid. The rest of the codons were found through this experiment. And here are my sources.